Hi. Good day. My name is Jhon Nichol from Crim23. This video is all about instructional/tutorial video about methods of identification other than fingerprinting.
Are you ready? Okay, let's begin. Anthropometry. What is anthropometry? Anthropometry or Yeshion system is developed by Alphonse Yeshion with the help from his father. He was able to successfully develop and implement anthropometry in 1882. This system consisted of using calipers and other measuring devices. Yeshion introduced his system of identification, which incorporates a series of refined, bodily measurements, physical description, and photographs. The Yeshion system was superseded by fingerprinting as the primary method of identification, though it remains an excellent means of furnishing a minutely descriptive portrait, valuable to investigation. Anthropometry is the science dealing with the measurement of the human body to determine difference in individual. These are the composition of Yeshion system. The Yeshion system is a technique for describing individuals on the basis of catalog of physical measurement, including standing height, sitting height, distance between fingertips with arms outstretched, and size of head, ear, foot, digits and forearm. In addition, distinctive personal features, such as eye color, scars, and deformities were noted. These are the example of Yeshion system or anthropometry. That is anthropometry or Yeshion system. Okay. Let's proceed to portrait parle. In 1882, Alphonse Yeshion, also called father of scientific detection, said that anthropology could be invoked to aid in the sight recognition and identification of criminals. Employing these methods of scientific description, he worked out a system of identification, utilizing 11 skeletal measurements which are practically unchanged after maturity and are not affected by increase or loss of weight. Portrait parle is a verbal and pictures description of a person being identified. Such information may be given by witness, relatives, or person who is acquainted with physical features of the person to be identified. Yeshion designed an incremental physical description card, also called the Yeshion card, comprising of 4 areas. Anthropometry, a field he enriched with new topological descriptions or ear, nose and iris. An incremental, a detailed physical description method which formed the basis of portrait parle. Photographic description, which he continually enhanced by defining a general protocol for face. An inventory and precise mapping of all specific marks to be found on the body, scars, tattoos, moles and the like. Forensic odontology. Forensic odontology is proper handling, examination, and evaluation of dental evidence which will be presented in the interest of justice. Forensic odontology is the application of dental science to legal investigations, primarily involving the identification of the offender by comparing dental records to a bite mark left on the victim or at the scene, or identification of human remains based on dental records. Forensic odontology is the branch of forensic medicine has established itself as an important and indispensable service in medical legal matters toward the creation of justice and secure society for the future inhabitants. Sex determination is very important subdivision of forensic odontology, which plays a major role in identification the unknown individuals in natural disaster. This is a chemical and nuclear bomb explosion scenarios. That is forensic odontology. There are 3 more methods of identification. The forensic anthropology, the forensic serology, and the DNA fingerprinting. Forensic anthropology. Do you want to know something about forensic anthropology? This is one of the interesting methods of identification. Forensic anthropology can be described as the analysis of human remains for medical legal purpose of establishing identity. Forensic anthropology, application of physical anthropology to legal cases, usually with a focus on the human skeleton. Forensic anthropology uses the techniques of physical anthropology to analyze skeletal, badly decomposed, or otherwise unidentified human remains to solve crimes. The role of the forensic anthropology focuses on applying detailed knowledge of the development, morphology, and variation of the human body as an aid to establishing, confirming, or indeed refuting personal identification. 4 basic biological criteria form the basis of any primary identification. A. Skeletal or developmental age. B. Biological sex. C. Living stature. D. Ancestry or racial affiliation. These characteristics are supplemented by secondary traits which include the following. Body modification. Evidence of surgical intervention, including scars and prosthetics. Evidence of trauma. Chronic hard and soft tissue diseases that may leave characteristic macroscopic and microscopic lesions on surviving tissue. And next is forensic serology. Forensic serology is the application of the study of blood, semen, saliva and other body fluids to legal matter. The use of blood in forensic analysis is a method for identifying individuals suspected of committing some kind of crimes. Serology is the study and examination of bodily fluids that is used in forensic science as a means of segregating fluids excreted by assailants or attackers in varying criminal acts. These acts can range from physical assault to sexual assault, right through to the act of murder, and all of them will have an element of fluid secretion attached to them. Serology allows the forensic scientists to segregate these bodily fluids when found at the scene of the crime and then perform a variety of tests on them in order to identify where these fluids originated from, or most importantly, who they came from. One important aspect of serology is determining whether or not stains resembling blood found at a crime scene are actually blood or some other stain that bears a similar resemblance. Types of serology. Serology is split into 2 categories of investigation. Presumptive testing. These tests provide 2 separate means of producing a result. One is to use compounds that can have an effect on blood when introduced to it. These results are a simple and quick way of proving that samples are actually blood, especially if time is of the essence. Confirmatory tests. This is a more involved set of tests that are carried out using samples of what is believed to be blood and mixing them with a chemical compound that reacts adversely with hemoglobin, the resultant factor being the production of crystals under the microscope that can be identified as blood. It is important that these tests, either one of them, are carried out to prove that these stains are in actual fact blood, and, more importantly, human blood. This is particularly important if the deceased's body has been found outside where it may be possible that animal blood has been spilled on the ground at some point. That is forensic serology. And the next is DNA fingerprinting. DNA fingerprinting is a method of identification that compares fragments of DNA. It is sometimes called DNA typing. DNA is the genetic material found within the cell nuclei of all living things. In mammals, that strands of DNA are grouped into structures called chromosomes. With the exception of identical twins, the complete DNA of each individual is unique. A DNA fingerprint is constructed by first extracting a DNA sample from body tissue or fluid such as hair, blood, or saliva. The sample is then segmented using enzymes, and the segments are arranged according to size using a process called electrophoresis. The segments are marked with probes and exposed on X-ray film, where they form a characteristic pattern of black bars, the DNA fingerprint. Other biological applications. Humans are not the only creatures who can be fingerprinted. Anything with a DNA or RNA genome, including animals, plants, bacteria, and viruses, can fingerprinted the same way. DNA fingerprinting thus has an incredible number of biological applications. The most common use of DNA evidence in courts is to help positively identify a rapist or murderer. This is done by comparing the DNA fingerprint of a sample of blood or semen left at the crime scene to that of the suspect. Fingerprints can also be made using DNA in urine, saliva, and any number of other bodily sources. Paternity testing. The DNA fingerprints of the child, the mother, and the father are compared. Paternity is determined by assessing whether it would be possible for the child to have inherited the combination of bands in their DNA fingerprint from the alleged father and mother. That is DNA fingerprinting. That are the 6 methods of identification. Thank you for watching. I hope you learned something.